Started? Okay. Welcome back, guys. Now, in this class, we'll be discussing about the genetics. Okay. We have completed the cell injury topic, and also we have discussed about the inflammation. Now, after this, we are going to discuss the third chapter. We are going to begin the third chapter, that is the genetics. Okay. Now, in this genetics, first itself I am telling you, see, what are the most important topics? In your exam, 100% there is no doubt there will be one question on the inheritance pattern. Whether it is autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive pattern, definitely they will give you one disorder and they will ask you what is the inheritance pattern. Or they can ask something like, so which of the following diseases follows X-linked recessive pattern? All of the following follows X-linked recessive pattern except. So such kind of questions can come in your exam. Okay. Having said that, now let's begin with the genetics. Now, look here. See, what exactly is a gene? Okay, so what exactly is a gene? Now you can very clearly see here, this is a cell. Okay, here is a cell. Now, inside the cell, we all know there is something called as a nucleus. Okay, there is something called as a nucleus. In the nucleus, what do we have? We are having chromosomes. We all have chromosomes, right? Humans have 46 chromosomes. Okay, 46 chromosomes, 23 from the father and the 23 from the mother. Okay, done. Now, if you look at one chromosome, now what exactly this chromosome made up of? So, if you unwind the chromosome, if you open the chromosome, what you are going to have is this material which is called as a chromatin. Okay, which is called as a chromatin. Now, even if you open the chromatin, okay, so what exactly is this chromatin, sir? What exactly it's made up of? The chromatin is nothing but some nucleosomes and the DNA. So, at the end of the day, the chromosomes are made up of this DNA. This is all the DNA, sir. Now, in this DNA, what do you have? Sir, in this DNA, the sequence, see here, this is the one strand of the DNA, this is the other strand of the DNA. The DNA, what it consists of? It consists of nucleotides. You can very clearly see here, these are the nucleotides. Now, a series of nucleotides or the sequence of these nucleotides, for example, from here till here, it will be one gene. Okay, so what exactly is a gene? So genes are present on DNA. Genes are nothing but a sequence. Okay, let me write. Sequence of nucleotides. Sequence of nucleotides, series of nucleotides is called as a gene. There are many genes. How many genes are there in the human body? How many genes are there? There are almost 20,000 plus genes. Okay, there are almost 20,000 plus genes. Now, in these genes, okay, for example, you are having many genes. Do you think all these genes are going to be working? No, most of the genes are not expressed. So, what are these exons, sir? MCQ in your MCQs in your exam. So, what are these exons? Exons are those genes which are expressed in a simple way. Okay, in a simple way we can say something like this. So those genes which are expressed, exon for expression. So those genes which are expressed. Okay, so those genes are going to be expressed in the sense they will undergo transcription, translation, protein synthesis. Okay, so exons are those genes which will undergo transcription, translation process followed by protein synthesis. Okay, so introns. So what exactly are the introns? Introns are those genes which are not expressed. They are not expressed. So they are present in our genome. We have exons and introns in our DNA, but exons are going to be expressed and introns are those genes which are not expressed. Okay. Now what do you think? Which will be more? Exons or introns? What will be more? Introns. Okay, introns are more in number. Okay, almost 98% are the introns. Okay, almost 88%, 98% of our genes are not expressed. Okay, now after this, let's discuss about the locus. See, first we are all starting with the basics. Okay, we are all starting with the basics from the chromosome level, from the chromosome, DNA, nucleus level. We are going one by one. Okay, what is a gene? How many number of genes are there? Where exactly the genes are present? Okay, now what is a locus? Locus, in a simple way, is a location of gene. The location of a gene is called as a locus. Now, if you ask me, so where are you staying? I have to give you my address, right? See, this is the place where I am living in this country, India. I am living in so and so state, in so and so district, in so and so town, in so and so street. Okay, so that's the exact precise location. Okay, now even if you want to know where exactly is the CFTR gene present, now I have to give you pinpoint location. So this is the chromosome on which this gene is located. So that location is called as a locus. Locus means the location of the genes on the chromosome. Okay. For example, you just look here. I'm giving you the example of CFTR gene. Okay. Guys, can you tell me where exactly is this CFTR gene is mutated, in which condition? There is this one condition where the CFTR gene is mutated. Guys, can you tell me what is that condition? Sardar Singh, can you tell me? Sardar Singh, fun mode. Where exactly we have studied about the CFTR? Not we haven't studied about the CFTR gene mutation. But do you have any idea where we have discussed where this CFTR gene is mutated in a condition called as Varun Kashyap? Excellent. Okay, that's the cystic fibrosis. Now look here, the gene is CFTR gene. Okay. Now where is the CFTR gene located? See, the location of CFTR gene is 7q31.2. So 7q31.2. Sir, what exactly is this 7q31.2? First, this letter number 7, it is denoting. Okay, this letter number 7, what exactly is this denoting? Letter number 7 means here 7th chromosome. On chromosome number 7, okay, first letter is chromosome. Okay, on chromosome number 7. Sir, what is this Q if you ask me? See, each chromosome have two arms, P arm and a Q arm. Okay, P arm and Q arm. P arm is the short arm and long arm is called as a Q arm. Even in this chromosome, you look here, the short arm is called as a P arm and the long arm is called as a Q arm. Okay, so the CFTR gene is present on chromosome number 7 on long arm. Okay, sir. Then where? See, this is all the long arm. Okay, now where, sir? Here. See, this 31.1. Okay, what is this 3 stands for? 3 stands for this region. Okay, region, sir. In the region number 3, see, 3 stands for what? 3 stands for region. And I, what is this? Sorry, not I, 1. 31, right? So what is this 1 stands for, sir? 1 stands for band 1. In this region, this is the first band. In the band 1 and subband 2. So this is the exact location of CFTR gene. So now tell me, what is this 7q31.2? 7q31.2 means first letter stands for, first number stands for chromosome, PRQ arm, next region, band, subband. So this is the exact place where the genes are located. Okay, they can ask you, they might give you, for example, in there is a syndrome called as Dye-George syndrome. Dye-George syndrome. Okay, Dye-George syndrome. There is a famous question. There is a deletion of chromosome number genes in 22q11.2. Okay, so there is deletion of genes from chromosome number 22q11.2. Now you know, sir, what is this 22q? 22 is the chromosome number, q is the arm, 1 is the region, 1 is the band and 2 is the subband. Okay, so this is the exact location, guys. Now after this, what else you should know? Okay, done. So locus, now you know it. So what exactly is an allele? So what exactly is allele, sir? Alleles are the alternate forms of the gene. Okay, alleles, they are the alternate forms of a gene. Now you can ask me, sir, these are like little confusing words like alternate forms of a gene, because students have a very limited knowledge, especially in their first years, okay, and second year, they have a very limited knowledge about the genes, chromosomes, alleles, okay, genotype, phenotype, these things. So what exactly is an allele? Hope already you have studied in your first year biochemistry. Alleles are the alternate forms of a gene, which means, dekho, you are having one gene. Okay, you are having a gene, sir. What is a gene? You already know it. It's a sequence of nucleotides. For example, imagine this is a gene. On the DNA, that particular segment is coding for a gene. Okay. Now, what it is coding for, sir? It's coding for the eye color. It's coding for the eye color. So, gene is one gene only. But, see, what are the different alleles? What are the different eye colors? Okay, the phenotype can be blue eyes, green eyes, grey eyes, okay, dark eyes. So these blue colors, the expression, the expressing thing, these are the different variables You are understanding? So now you can have a one gene. Yes, gene is one, but these two are the alleles. You know it. Black eyes, blue eyes. Not only just that. For example, you look here. This is, imagine this is a gene. So one gene is having how many alleles? See, there can be many number of alleles. Most of the students think there are only two alleles, sir. No, there are not just two alleles. There can be many alleles. Okay. For example, you can have the gene is one gene, the gene for eye color. This is a eye color gene. Okay. Now, this eye color gene, it may be coding for, maybe coding for dark color or blue color, green color or gray color eyes. Okay. Variations. Next, for example, here is a gene for hair color. It can code for different color hairs. Okay. So, a gene is having multiple alleles, different variables, alternate forms is called as a allele. For example, let me tell you. See, gene. First, I will give you a gene. What is that gene for? You should tell me the alleles. Okay, you should tell me the different forms. Now, height gene. Gene for height. Height of the person. Now, can you tell me what are the alleles possible? What are the basic? Okay, what are the alleles which you know? Sir, if height is a gene, how many alleles it can have? Guys, can you tell me? Come on, guys. Sumit, Pashwan, Reen. If height is the gene, if height is the gene, how many alleles? See, one thing is it can code for tall height or it can code for short height. Possible, tall and short. Simple. Okay. So, if for example, let me give you one more example. If gene is for skin color. If gene is, okay, for the skin color, what are the different alleles possible for the skin color? White color skin or black color skin or dark color skin, brown color skin, there are different colors. So these colors, these variables are called as the alleles. Okay, now we understand what is the difference between the gene and alleles. Gene is what it is coding for. Alleles may be different. Okay, different forms. After that, now the next term which you should know for your basic understanding at this level is homozygous and heterozygous. Later you will understand the patient is homozygous for so and so. The patient is a heterozygous for so and so gene. So what exactly is this homozygous and heterozygous? Okay, let me give you some example. Imagine my alleles, for example, look in me. For a height gene is height. Now for this height gene, how many alleles are possible? 2 alleles are possible. What are they? Tall and short. Okay, tall and short. Okay, broadly, just generally broadly classifying a tall, short. I'm just leaving the intermediate like an average and just taking it out. So tall and short. So for tall, I'm just representing it as capital T. Capital T is the allele for tallness. For shortness, let's take it as a small t. Okay, small t. So capital T is the gene for tallness. Okay, tall. And small t is the gene for shortness. So what is homozygous? Imagine in me. See, if there is a person who is 6 feet tall, okay, now I am telling you, he is having capital T and capital T. What does this mean by capital T, sir? Capital T. It means, dekho, now this individual, imagine, see, there is this guy, okay, who is 6 feet tall. Now, tell me, he is having this gene, sir, height gene. How many copies of this height gene can be present in him? 2 copies, one from the mother and one from the father. Okay, one from the mother and one from the father. Excuse me. One from the mother and one from the father. See, both from the mother as well as from the father, now he is receiving both the genes, sorry, I beg your pardon. Both the genes are coding for tallness. So he is homozygous. Why I am telling it's homozygous? Because both are capital T, capital T. Now, for example, if I am the person, okay, who have received one capital D gene from the father, which is coding for the tallness, and one small t from the mother. Now, the genetic makeup will be capital T, small t, capital T, small t. Now, is this person homozygous or heterozygous? This is heterozygous. Why? Because from the father, capital T is coming. From the mother, small t is coming. See, the genetic makeup, simple, what is homozygous and what is heterozygous? In the genetic makeup, if you have same type, if you have same type of alleles, then it is called as a homozygous. If you have two different types of alleles, one capital T, one small t, then it is called as heterozygous. Now, I will give you one example. Can you tell me? Imagine there is this one person, okay, there is this one person who is having his genetic makeup something like this, small t, small t. Is he homozygous or heterozygous? What do you think? Is he homozygous or heterozygous? Small t, small t. This is definitely homozygous. Which means, what exactly this mean by? He got two alleles, one from the father and one from the mother. Both are coding for short only. Okay, so this patient is going to be definitely short. Okay. Next. After this, what else you should know? You should know what is this dominant allele, sir, and what is this recessive allele. A dominant allele, what exactly is this? And this recessive allele, what exactly is this? Okay. Look, dominant alleles, for example, now I am the person, imagine I am the person who is receiving capital T from the father and small t from the mother. Okay, capital T is coding for tall height and small t is coding for short height. Okay, now in both of them, who is going to be expressed? See, this capital T, it's going to be expressed. Okay, it's a capital T, it's going to be expressed. Out of these two, see, one is coming from the father and other is coming from the mother. But still, only one gene is going to be expressed. Okay, they will decide. Okay, out of these two, I am telling you, see, this capital T is a dominant gene because it is expressing. So, dominant alleles, out of these two alleles, the one which is expressed, the one which is going to undergo transcription, translation, final expression is called as a dominant allele. Okay, so recessive alleles are not expressed usually when in a heterozygous state. Okay, now, dekho, recessive allele, what exactly is a recessive allele? Usually they do not express. But when they will express in homozygous state. Okay, they will express in homozygous state. So, what exactly is this, sir? Let me give you one example. Look here, small t, small t. Okay, small t, small t. So, what exactly is this small t, small t? So, small t, it's a recessive allele, right? It's a recessive allele. This small t is also a recessive allele. Now they will express. But in this condition, whenever there is a capital T, that is a dominant allele, now it's not going to be expressed. So now tell me, in this condition, the patient is going to be tall or the patient is going to be short? First of all, you know that's a heterozygous state. I am the person who is having one capital T allele, one small t allele, one from the father, one from the mother. So now in this condition, tell me, am I going to be tall or am I going to be short? What do you think? Make it faster. Understood this concept? Chahar, yeah, you might able to understand, but I have to consider everyone, right? So that's why I have to build from the basics. Okay. Now tell me, just answer this. This patient is going to be tall or this patient is going to be short? This patient is going to be absolutely tall. Why? Because the recessive allele, yes, the recessive allele is not going to be expressed. Now, let's see some examples. Okay, let's see some examples. Now, this is, for example, A and B. Now, here we are having a, for example, male. This is a female, male and female, father and the mother. Now, father is having brown eyes and mother is also having brown eyes. Now, father is having, everyone will have two alleles. Okay, father is also having two alleles. One capital B, one small b. Mother is also having one capital B, one small b. Okay, now right. Now tell me. So, father and mother both are homozygous or heterozygous? Both are heterozygous. See, both father and mother both are heterozygous in their genes. Now, whenever they have children, whenever they have some children, now combinations, how can it can be possible? Capital B, one capital B, one small b. Okay. Now, one small b, one capital B. And see, there is also a possibility that from the mother, the small b is coming. From the mother, the small b is coming, and from father, the small b is coming. And now see, whenever the recessive genes, whenever the recessive alleles, whenever they are in homozygous state, only then they are going to be expressed. But in all the other conditions, look here, capital B, that is homozygous, but both the alleles are dominant. So definitely brown eyes. Here also see, this is heterozygous state. Now this child is having one capital B allele, one small b allele, but he is having one dominant allele. So because of this B, capital B, the child is going to have dark eyes. Okay. Now here also same thing. Okay. I hope you can understand this. Same. Look here. Now only question which I want you to know is, tell me, father is homozygous or heterozygous? The father is heterozygous, but mother is homozygous for the recessive alleles. Father is heterozygous, one dominant allele, one recessive allele. Now in this condition, what happened? See, BB means when recessive alleles are in homozygous state, now they are expressed blue eyes. Okay, so this is the concept which I want you to know. Now after this, the next topic which I want you to know for your exams is the codominance. So far you have said that whenever there are two alleles, one dominant allele, one recessive allele, only dominant allele is going to be expressed, the recessive allele is not expressed. Okay, but when you have two dominant alleles, okay, when the two alleles are dominant, what happens? Who is going to be expressed? Both will be expressed. Okay, when both the alleles are dominant, both will be expressed. So this is called as a codominance. Okay, combination. Okay, codominance. Both are going to be expressed. Now look here. What is codominance? Both dominant alleles are expressed. Both the dominant alleles are expressed. Example is, this is the one important for your exam. ABO blood grouping and HLA genes. See, this ABO blood grouping and HLA genes, they follows this codominance pattern. Okay. Now in this table, if you look, see, these are the alleles from the father. These are the alleles from the mother. See, for blood grouping, how many alleles are there? First you need to understand. For tallness, for height, how many alleles are there? There are two alleles. That is capital T, small t, right? In the same way, for blood grouping, there are three alleles present, sir. Three alleles present. What are these three alleles? A, B, O. Okay, and even in the father, there is A, B, O. Now, when this mother and father, when they give rise to children, okay, when they give rise to children, what are the combinations possible? Once look here, sir. This A and A, when the father is A and mother is A. Okay, what can be possible, sir? Now, the children will be definitely A. Okay, the children will be definitely A. Okay, now, father is A blood group, mother is B blood group. Father is A and mother is A. So, now see, what I want to put into your mind is, look here, this A and B alleles, A allele, B allele, both are dominant alleles. Both are dominant alleles. As both are dominant alleles, in the baby C, they are having AB blood grouping. It's not A or it's not B, it's both AB blood grouping. Okay. Now, for example, A and father is A and mother is O. Okay, father is A and mother is O. Now, O is always recessive. Okay, O is recessive. A is dominant, B is dominant, O is recessive. Now, in this condition, now what the baby is going to be? Sir, you know, only dominant allele express. Recessive alleles do not express. So, when this father and mother, when they have this child, father is A, mother is O, the baby is going to be definitely A. Okay, now this baby's genetic makeup is A-O, A from the father, O from the mother. Only A is going to be expressed, O is not going to be expressed, A blood group. Okay, simply you can follow the table. Now, only point which I want you to know is, whenever there are two alleles which both are dominant, both will be expressed. This is seen in conditions of ABO blood grouping as well as HLA genes. Answer. Okay. Now, after this, next topic which we have to discuss is the genotype. So, what is genotype and what is phenotype? Okay, what is genotype and what is phenotype? Any idea, guys? What is genotype and what is phenotype? Kashyap, from our side, from our end, the stream is absolutely all right. Okay, there is no issue with the stream. If there is any lagging or buffering, it's from your side. Okay, it's from your internet. Like, there is some internet issue from your side. Okay, not from our side, sir. So now, what is a genotype? Any idea? See, genotype and phenotype, most of the students confuse this here. Genotype, let me tell you, first of all, look here. Genotype is the organism's genetic makeup. Okay, the organism's genetic makeup. In a simple way, we can write here, genetic makeup of an organism. Okay, genetic makeup of organism. You will understand better with the examples. Now, what is a phenotype? Phenotype is what you can see, what is getting expressed. Okay, how are the genes? Is the genotype. Okay, what is the composition of the genes? Like, let me give you, for example, see, genotype versus phenotype. The genotype means, genotype of an organism is defined as actual complete genetic makeup of an organism. It refers to what? It refers to the pair of alleles, whether it is capital T, small t, or one capital T, one small t. Okay, whether they are homozygous, heterozygous, about the genetic makeup, how the genes are and how the alleles are. If you are talking at the level of alleles, then it is called as a genotype. Okay, now for example, see, now I am saying I am having one capital T allele, one small t allele. See, this is the genotype. We are talking about the alleles, how the alleles are. And what is a phenotype? Now, because of as I am having one dominant allele and small allele, what is the phenotype? The result, the expressed one that you can see from outside. It is tallness. This tallness is the phenotype. Now, for example, see, this is the genotype. I am homozygous for recessive allele, small t. Then what is the phenotype? The phenotype is short. The phenotype is short. So, this is the genotype. The genetic makeup is called as a genotype. What you can see from outside is called as a phenotype. Okay, the expressed things. Now, let's once go through this. Genotype of an organism is defined as complete genetic makeup of an organism. It also refers, look, it also refers to a pair of alleles inherited in an individual for a particular gene. That's a genotype. See, at the level of genetics, at the level of genes. Then what is phenotype? Okay, whether the type of blood, AB blood grouping, the hair color, the eye color, the skin color, the height of the person, what is getting expressed. Phenotype refers to an individual's observable traits such as height, eye color, blood type. Okay, that's what. Okay, so phenotype is determined by, tell me, phenotype, it is determined by the genotype. It's the genes that determine the phenotype. It's the genes that determines what is getting expressed. Okay. So, even here in your class 11th and 12th you would have studied. See here, BB. See, this is, you can definitely say this is homozygous and capital means dominant. See, this is the genotype. Because of this genotype, there are purple colors. Purple color flowers you would have studied in your, just for easy understanding, I am just explaining this. In class 11th and 12th you have studied the Mendel's experiments on pea plants. Okay. Now, small b means homozygous recessive. Whenever there is homozygous recessive, what will be the phenotype? The phenotype will be white colors. This is the phenotype. This is the genotype. Okay, done. Now, after this, let's explain you about the mutations. Guys, can you please tell me what are the mutations called as? What exactly are the mutations? What exactly are the mutations? Can you tell me? Any idea, guys? Sardar Singh, it's not just actual appearance. See, there are enzymes in our body. You cannot see the enzymes, right? Okay, there are proteins in the body. You cannot see the proteins. So, it's not just what is expressed to the eye, how the genes are getting expressed and what is the resultant product. You cannot see an enzyme or you cannot see a protein. Okay, but they are also getting expressed. So, phenotype is what is getting expressed, what type of protein getting expressed. Okay, but in a general understanding, you can say, what is the appearance? Yes, that's correct. Easy understanding. Okay. Now, what are the mutations? Any idea? Permanent. change in DNA material. A sudden permanent change in a DNA material is called as a mutation. A permanent sudden DNA change. What might be the reason? I might be exposed to ionized radiation, X-rays, or I might be exposed to some, like, in the nuclear area, there is this area where the nuclear explosion have happened. Now, if I am the person who is in that area, because of the extreme radiation, that radiation can cause damage to my DNA material. So, damage to the DNA material, which is permanent, is called as a mutation, irreversible in nature, sir. Okay. Now, for your exam, what you should know is point mutation. How many types of mutations are there? There are two types of mutations: point mutation as well as frame shift mutation. Point mutation and frame shift mutation. So first, what exactly are these point mutations? Okay, point mutations, what exactly are they? Any idea, guys? Fun mode? Yeah, permanent heritable change. Permanent heritable change, which means these changes, they can also get inherited to the offsprings. Okay. Today I'm little cold. Got cold. So, do you have any idea about the point mutations? See, let me tell you, point mutation means, look here, this is normal gene. So, DNA, in the DNA, here is a normal gene present. Okay. Now, in this normal gene, everything is okay. You can very clearly see here are the nucleotides, thymine, adenine, cytosine, thymine, guanine, cytosine, adenine. The sequence of the nucleotides are there. Okay, good. Now, what happened is point mutation. Now, see, in this area, this T is replaced by C. Okay, the nucleotide, one nucleotide is replaced. Now, instead of T here, now it is replaced with C. So, it's just at one particular point, right? Not the entire chromosome is lost. The entire chromosome is not lost. Okay, only in that particular point, there is replacement of one nucleotide by other nucleotide. So, it is called as a point mutation. Okay, so what happens if there is a point mutation? Okay, let me tell you what happens if there is a point mutation. Three possibilities are there. Okay, if there is a point mutation, there are three possibilities. Before going to the possibilities, let me tell you an example. Look here, the example is sickle cell anemia. Yes, okay, sickle cell anemia, sir. Normally, okay, wild type means normal. Okay, normal hemoglobin. This is the gene which is coding for the normal hemoglobin. Now look here, there is CTC. Sir, I am right now talking at the level of DNA. I am right now talking at the level of DNA, sir. The genes are present in DNA. Now what happened is, say, this is normal hemoglobin, CTC. Whenever there is CTC, first transcription will happen, right? First transcription will happen. So from this DNA, who is going to be synthesized? mRNA. mRNA will be synthesized. Dekho, this is the mRNA. Now, whenever here on DNA, whenever there is CTC present, now on mRNA, who will be present? Sir, C in the place of C, see G, A, G. Okay, wherever there is cytosine, on the opposite end, guanine will be present. Wherever there is thymine, adenine, and wherever there is cytosine, there will be again guanine. Okay, this is something normal. Okay, on mRNA, what you are having? GAG. See, this GAG, this is a trinucleotide. Okay, this is a codon. This GAG is the codon, which is normal. Now, this GAG codon is coding for what? It is coding for glutamic acid. So, in the hemoglobin, there is glutamic acid present. Okay. Now, what happened in sickle cell anemia? Point mutation. What is this point mutation? See, in the place of T, who came? A came. In the place of T, the thymine is replaced by adenine. Now, because of that, whenever there is a change in DNA, automatically there will be change in mRNA also. So, in the place of A, okay, see, whenever there is A, who will sit? Uracil will come and sit here, uracil. Okay, why? Because adenine, wherever there is adenine, the opposite, the complementary, you will be having uracil. Okay, you will be having uracil in the mRNA. Okay, so instead of A here, you are having now uracil. Now what? Sir, now this codon, this GUG, this codon, sir, this trinucleotide, Now this trinucleotide, this codon is going to code for valine. So in the place of glutamic acid, okay, the glutamic acid, now it is replaced by valine. So this is normal hemoglobin. So can you call this as a normal hemoglobin? No, this is not normal hemoglobin. So this is sickle cell hemoglobin. So now you have to tell me, sir, in sickle cell hemoglobin, what happened? Now the glutamic acid, okay, the glutamic acid is replaced by valine. The glutamic acid is replaced by valine because of a point mutation in the DNA. Whenever there is a point mutation in the DNA, there will be a change in the mRNA. Whenever there is a change in mRNA codon, automatically it codes for a different amino acid. That will cause change in the proteins. Now hemoglobin is getting affected here. This is a sickle cell hemoglobin. Okay. So done, sir. After this, what else you should know? See, because of these point mutations, what are the possibilities? The three possibilities are there. Okay, point mutations. Can lead to. Okay, point mutations are of three types. Okay, I should say point mutations are of three types. Before discussing they can lead to, point mutations are of three types. What are they? First type are called as silent. Silent mutation. Second one, nonsense. Oh, before telling you nonsense, first let me write missense. Missense mutation. And third type are called as a nonsense. So now tell me, what exactly are these? Sir, point mutations are of three types. Point mutations by themselves, there are three types, sir. What are they? Silent mutations, missense mutations, and nonsense mutations. Okay. So first of all, let's discuss about what is a silent mutation. Okay. So what is a silent mutation? Look in this example. Okay. Look in this example. Sir, normally it should be GAA on mRNA. Okay, on mRNA, it should be, the codon should be GAA. If the codon is GAA, then it codes for which amino acid? Glutamic acid. It's coding for glutamic acid. Now what happened is point mutation, one small point mutation in the DNA. So that in the place of adenine, now who came? Guanine came. It was supposed to be GAA. Now in the place of this A, now who came? GAG. Now, but still you should understand certain things. One amino acid, dekho, one amino acid, same, glutamic acid. One amino acid is coded by many codons. There are many codons, okay. Like for example here, in this case, GAA is coding for glutamic acid, even GAG, that codon, it will also code for glutamic acid only, okay. One thing, never ever forget, one codon codes for one amino acid, that's it. One codon codes for one amino acid, but one amino acid have multiple codons, okay, done, sir. But here, because of the mutation, A is replaced with G. A is replaced with G, but still amino acid is, I beg your pardon, but the amino acid is still the same, glutamic acid only. So if there is any problem now, will any problem occurs now? So there is no problem. So this type of mutations are called as a silent mutation. Mutation occurred, point mutation occurred, but still there is no issue because it's the same amino acid. So this type of mutations are called as a silent mutation. Next, sir, next. Dekho, this is the codon, GAA. These three nucleotides are called as one codon. GAA, this is called as a one codon. Now this GAA, it is coding for what? It is coding for glutamic acid. It is coding for the glutamic acid. Okay, well and good. But some point mutation occurred in the DNA. So in the RNA, see, it was supposed to be GAA. Okay, the codon should be GAA. But now what is happening in the RNA? Now, in the place of G, U is coming. Okay, in the place of G, U is coming, uracil is coming. In the place of guanine, uracil is coming. So whenever you are having UAA, this is called as a stop codon. So immediately the protein synthesis stops. Translation stops. So these are codons. Okay, UAA, these are the codons which will stop the process of translation. Immediately translation will stop, sir. So this type of mutation where a stop codons are getting produced because of the point mutation. Now it's a stop codon. So they are called as nonsense mutations. So nonsense mutation means there is, because of the mutation, now you are end up with a stop codon. Now in your exam definitely they will ask you, what are the stop codons? Which codons are responsible for stopping the process of translation? Okay, what are they? UAA, UAG and UGA. Okay, see these three codons. Okay, in the entire world there are these three codons which will stop whenever you are having this codon in the mRNA. Whenever you are having this codon on the mRNA, see, translation is happening with the help of the ribosome is doing translation. Proteins are synthesizing. Now, whenever this ribosomes, whenever they comes to this UAG, UAA or UGA, immediately protein synthesis will stop, sir. Okay, so this is called as nonsense mutation. Silent mutation I have explained. Point mutation leading to what? No problem. There is no problem because same amino acid is produced. Next, nonsense mutations, stop codons. And what is a missense mutation? What is a missense mutation? Now, see, it was supposed to be GAA. Okay, it was supposed to be GAA. It is coding for glutamic acid. Now, in the place of adenine, cytosine is coming and sitting in the mRNA. Now, it's a different codon, totally different type of codon. It's not GAA, it's now GAC. Now, whenever there is GAC, it codes for what? It codes for a different amino acid. Which amino acid? Aspartic acid. So, instead of glutamic acid in the protein, now there is aspartic acid. So now this is not a normal protein. This is definitely a defective protein. So this type of mutations are called as a missense. Okay, missense mutations. Okay. Now you should tell me this example of sickle cell anemia. It is point mutation, okay, but is it nonsense? Is it silence? Or is it missense mutation? Now tell me. Guys, tell me. The sickle cell anemia, there is replacement of glutamic acid by valine. So it is an example of which type of point mutation? Missense, nonsense or silence? Fun mode, Sumit and made ECG, yes, you are true. Okay, so it's an example of missense mutation. Done, sir, done. Okay, so now just for me, tell me how many types of point mutations are there? There are three types of point mutation. What are they? Missense, nonsense and missense. Missense, nonsense and silence. Okay, done. After this, let me discuss about the frame shift mutation. So frame shift mutation. So what exactly are these frame shift mutations? Now look here. Now imagine there is this mRNA, sir. Okay, transcription completed. Genes are perfectly normal. Okay, for example. Now, okay, let me put it this way. So now, see, imagine this is a DNA. Okay, here, imagine here is a DNA. Okay, sir. Now, in the DNA, what do you have? You are having amino acids. Sorry, not amino acids, nucleotides. Okay, in the DNA there are nucleotides. See, imagine these are all the nucleotides. Okay, nucleotides sitting like adenine, guanine, cytosine, thymine. Okay, adenine, guanine, cytosine, thymine, same. This is the DNA, imagine, DNA. Now, from the DNA, who will come? So, from the DNA, mRNA is going to be produced. Okay, mRNA is going to be produced. Now, in the mRNA, what do you have, sir? The complementary things, right? Complementary things. Now, here also same. Now, on the mRNA, now here also you are having the nucleotides. Okay, these are all the nucleotides coming. Now, what happened is, normally, we will take 3 nucleotides, right? 3 nucleotides is considered as one codon. 3 nucleotides is considered as one codon. Okay, this is something normal. 3 nucleotides is considered as one codon. Now, what happened? Sir, one of the nucleotide, okay, here, now at the level of DNA, I am talking at the level of DNA. Dekho. Now, one nucleotide you are adding here. Now, there is addition of a nucleotide. Now, whenever one nucleotide is added, same in mRNA also, one nucleotide will be added, no doubt. Okay, now, dekho, what is happening? Now, one nucleotide is being added here. New nucleotide. Now, what happens? Now, these 3 will become the codon. Now, these 3 will become the codon. So, entire entire codons, there is a shift. Okay, now entire protein is going to be affected. So just because insertion of one nucleotide extra, just because of the insertion of one nucleotide extra, and the same way deletion also, addition or deletion can completely shift the frame. We can completely shift the frame. So let me give you a better example. Look here. Now I am going to discuss about the frame shift mutation. Clearly see here, frame shift mutation. Okay. Now, here are the codons. See, this is the mRNA. Now in the mRNA, adenine, uracil, guanine, adenine, guanine, uracil. Now you know it. 3 nucleotides are considered as one codon. See, these 3, AUG is coding for methionine. AAG is coding for lysine. UUU is coding for phenylalanine. Okay, amino acids. Okay, good. But what is happening? Okay, what is happening? See, when a base pair is inserted, now insertion I have explained you, one base pair, one nucleotide, it is inserted, sir. Okay, now see, extra U, it should be what? It should be AUG. Now, what happened after AUG? See, after AUG, one base pair is inserted. Now, first is AUG and second one is AAG. Now, look here, this extra U is added. Now, what had become? Now, first, okay, no problem. See, AUG is coding for methionine, but because of insertion of this extra U is coming here. Now, these 3 will become the codon UAA. Now, you know it. UAA is what? UAA is a stop codon. So, immediately what is going to happen? Translation is going to be stopped. Okay? Translation is going to be stopped, sir. Now, what I am trying to put into your mind is, if you add a base pair or if you delete a base pair, what might happen? The entire reading frame is going to shift. See, this is called as a frame shift mutation. Now, you can look here very clearly. Okay, you can look here. Sir, base pair deletion. If you delete one base pair. Okay, if you delete one base pair. Now, see what happens. Normally, what it should be? Just look here. It should be AUG. First 3 should be AUG, AAG. After that, UUU. Look here. It should be UUU. One U is removed. For example, if I remove one U, that's what I am doing, sir. One U is removed. If you remove one U, first AUG is coding for methionine. Okay, sir. AAG is coding for lysine. Okay, but it should be UUU. You have deleted one U because of some mutation. It's gone, sir. Because of some radiation or something, it's gone. Because of some chemical, because of some toxin, it's gone. Okay. Now, this will become the codon. UUG will become the codon. Now, UUG code for different amino acids. Now, it is coding for leucine. It was supposed to be phenylalanine here. It was supposed to be phenylalanine. Now, in the place of phenylalanine, leucine is coming. Frame shifted, right? Now, see, it was supposed to be, after UUU, it was supposed to be GGC. GGC should code for glycine. Now, what is happening here? Because of the shift, now it became GCU. Now, this GCU is code for total different amino acid. Entire protein structure is going to be affected, sir. Okay. So, what I am trying to put into your mind is only one thing. Point mutations are of 3 types. What are the 3 types of point mutations? The 3 types of point mutations are silent mutation, missense mutation, and nonsense mutation, and frame shift mutations. Okay, the second type of mutations which I want to discuss here are the frame shift mutations. See, these frame shift mutations, the frame shift mutations are because of either addition of a base pair or a deletion of a base pair. The entire reading frame will be shifted, which will cause damage to the protein, which will cause entirely different amino acids going to be present in the protein, which will cause the damage to the proteins. Okay, entire protein will be affected, sir. Defective protein will be formed. So done. Now after this, let's discuss about genetic disorders. Okay, now let's discuss about some genetic disorders. Is that okay, guys? Okay, now let's discuss about some genetic disorders. Now, the genetic disorders are mainly classified into 4 types. What are they? Mendelian, those genetic disorders which are following the Mendelian inheritance. The Gregor John Mendel, he gave certain laws, he proposed certain laws. This is how the diseases are going to be transmitted. This is how the genes are going to be transmitted from one parent to offspring. Okay, so Mendel's laws of inheritance. So those disorders which are following the Mendel's laws of inheritance. Okay, autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive. See, the point which I want to put into your mind is all this autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive, and even Y-linked diseases. They all follow Mendelian laws of inheritance. And these are the Mendelian disorders where there are point mutations. In all these conditions, there are point mutations. Okay, in almost most of them, there are point mutations. Okay, there is one base pair replacement. Now, there are certain diseases which do not follow Mendel's laws of inheritance. These are special. They do not follow Mendel's laws of inheritance. We will discuss. And there are chromosomal disorders, not just the genes, the entire chromosome is deleted, or half of the chromosome, Q arm is deleted, or there is mixing between two chromosomes, chromosomal disorders. For example, Down syndrome. What exactly is Down syndrome? Sir, Down syndrome is trisomy 21. Okay? I beg your pardon. Trisomy 21, which means it's a chromosomal disorder. One extra chromosome came. Turner's. What exactly is Turner's? 45X0. X0, 0 stands for one chromosome is lost. X chromosome is lost. One X chromosome is lost. So they are the chromosomal disorders. Turner's, Edwards, Patau, Klinefelter's. Okay, these are all the chromosomal disorders. Now, multifactorial disorders. What exactly are the multifactorial disorders? Do you guys have any idea what are the multifactorial disorders? What are multifactorial disorders? Multifactorial disorders means there are diseases which just do not depend on the genes. They are not just dependent on the genes, they are dependent on the multiple factors. Okay, they are dependent on the multiple factors. For example, hypertension. Okay, hypertension, sir. See, hypertension, it is not only dependent on genes but also on the other factors like, sorry guys, I'm having cold. So hypertension is the one thing which is not only dependent on the genes. Yes, of course, it is a familial, yes, possibility is there, but the patient's lifestyle, how much salt intake, the patient's food habits, the patient's lifestyle, smoking, alcoholism, or the patient's stress, the kind of environment he is working in, they are all going to determine whether the patient is going to develop hypertension or not. Okay, so a disease or a disorder, hypertension, it is dependent not only on the genes but also on other factors like lifestyle modifications, lifestyle, the food habits, the kind of environment he is working in. Okay, so these are the multifactorial diseases. Now, in your exam, the question that will come is, which of the following diseases? Which of the following diseases follows? Which of the following diseases follows multifactorial inheritance? Okay, which of the following diseases follows? Yeah, thank you for mode. Which of the following diseases follows multifactorial inheritance? Now, can you tell me? This is the question. Multifactorial inheritance is followed by diabetes mellitus, hypertension. Okay, diabetes mellitus, hypertension, cleft lip, and cleft palate. This is the question that was asked in the PG exams. So, which of the following diseases have multifactorial inheritance? Diabetes mellitus. See, diabetes mellitus is not only dependent on the genes. If the patient is sedentary, he is not working, he is taking excessive amount of sugars, more chances that he will get into the diabetes. Okay. Now, even though the patient is having certain genes for the diabetes, which are mainly a risk factor for diabetes. See, imagine I am the person who is having the genes which can take me to the diabetes, but with the kind of lifestyle I am having, okay, I can, okay, with the kind of lifestyle that I am having, I can go into diabetes very quickly. For example, if I am a sedentary person, I am taking excessive amount of sugars, I am not doing exercise, I am taking all kind of street foods, I will get into diabetes more quickly. Okay, so that's what I mean by. Now, anyway, after this, let's discuss about the autosomal dominant disorders. But wait, just tell me, how many types of genetic disorders we are going to discuss in this chapter? We are going to discuss about Mendelian disorders, Autosomal dominant, autosomal recessive, X-linked dominant, X-linked recessive and Y-linked. Here, in your PG exam, this was a question asked, Y-linked inheritance pattern. Okay, Y-linked inheritance pattern is also called as Hollandric inheritance. Okay, so Hollandric inheritance or Y-linked inheritance. That's the question which I want you to know. Next, autosomal dominant. See, these autosomal dominant disorders, what exactly are they? See, let me give you examples later. First, let me tell you basic things. The most common mode of inheritance out of X-linked dominant, X-linked recessive, autosomal dominant, autosomal recessive and Y-linked, out of all this, what is the most common inheritance pattern? The most of the diseases follow autosomal dominant pattern. Now, in this autosomal dominance, first you need to understand the concept of penetrance. So what will be the penetrance? The penetrance here is incomplete. There will be incomplete penetrance. Okay, see, these disorders, they will have incomplete penetrance. And even in these conditions, the expressivity in autosomal dominant condition is going to be variable or wide expressivity is seen. Now, most of the students, they don't have any idea. They don't have a proper idea about what exactly is the penetrance and what exactly is the expressivity. Okay, let me give you examples. First, see the definition. Later, I will give you the example. With that example, you will have the clear-cut idea. You will get a clarity. Now, look, what is a penetrance, sir? Penetrance means, okay, you know, in autosomal dominant disorders, the penetrance is incomplete penetrance. The expressivity is also incomplete. Wide variable expressivity is seen. Which means, first, let's discuss about the penetrance. Sir, penetrance is how often does someone with the genotype actually shows the corresponding phenotype. Okay, how often those people with a particular type of genotype will show the corresponding phenotype. Looking little complicated, right? Okay, looking little complicated, right? For example, look at this example. Okay, for example, look at this. This is a very funny example which I have find it out in the internet. Now imagine you are doing some experiment. Okay, you are doing some research. Okay, on 100 people. Okay, on 100 people. Now in this 100 people, you came to know that there are this 18. Okay, there are 3 rows, 6 in each. These 18 people are having a mutation. They are having a mutation, particular homozygous mutation. For example, capital R. They are having mutation. Okay, for this gene, particular gene, they are having a mutation. Okay, now see, these 18 people are having mutations. Sir. All these 18 people are having the same mutation. Okay, now, okay, let me put it this way. YRR, easy for easy understanding, you can say they are having this mutation. Okay, now, see, dekho. In these 18 persons, what exactly is happening? Look at their ears. Now, it's a mutation for the ears. Now, some of these people are having a very big rabbit kind of ears, big ears, or rat kind of ears. Okay. Now, tell me, in all these peoples, they are having the same mutation, but look, everyone is getting affected. No. See, these people, they are having normal ears. There are these people who are not having normal ears, sir. So, in all these people, same mutation is there, same homozygous mutation is there, but all of them are not having the disease. All of them are not having the same phenotype. So, this is called as a penetrance. Now, in this condition, if everyone is affected, I will say penetrance is complete penetrance, I would have said. But in this condition, dekho, the penetrance is not complete. The penetrance is incomplete because even after having a defective gene, still the patients are normal. Some of the patients are normal. So, now look at this definition. How often does someone with a genotype, okay, how often does someone with the same genotype, these are all the people with the same genotype, shows corresponding phenotype? Okay, now look here. Example, sir, out of 18 people, 11 people, see, out of 18 people, 11 people are showing the phenotype. Okay, they are all having this big ears. So, how much is the 11 out of 18 are affected. Okay, even though after having the same genes, 11 out of 18 are affected. So what is the penetrance? Penetrance is 61%. Incomplete penetrance. So such a type of penetrance is shown by autosomal dominant disorders. If the patient is having autosomal dominant disorders, then the penetrance, even though the patient is having some defective genes, still they do not express the disease. This is called as penetrance. Now after this, look at the expressivity. Look at the expressivity. So what exactly is expressivity, sir? Expressivity means, okay, this is our expressive. Okay, expressivity. Expressivity means variation, dekho, variation in severity. Variations in the severity are expression of a phenotype. These are the two definitions you can understand. Expressivity means variations in the severity. Now, in this example, dekho, sir, yes, you know, the 16 individuals having the same type of genetic mutation. Even though after having the same type of genetic mutation, only 11 people are showing the disease. So penetrance is 61%. Penetrance is incomplete. But even look at the affected individuals. Sir, these affected individuals, see, okay, these affected individuals, now in this condition, look, in this example I am giving, these are the affected individuals. Now, are they having the same size ears in this example? Yes, sir, they are all having the same size ears. Dekho. So, what is the expressivity? Sir, here the expressivity is wide range of expressivity is there? No, there is a narrow range of expressivity. All are having the same type of expression. All are having the same type of phenotype. Sir, you can say here gene mutation is there. All the patients who are affected are having same type of phenotype. But look here in this example. Sir, in this example, look, that this patient is affected having a big size ears. Now, these patients are affected who are having small size ears. Okay, so even after having same type of genetic mutation, even after having same type of genes, the expression, the phenotype is different. Different types of phenotypes are getting expressed. Okay, so this is called as variable expressivity or variable expression. Okay, variable expression. Now, let me give you one example. For example, imagine, simple example I will give you. Diabetes mellitus. Okay, diabetes mellitus. Of course, it's a multifactorial inherited disease. Now, diabetes, if I am the patient who is having a diabetes and there is some other patient who is having a diabetes, do you think, see, we are going to have the same kind of clinical features exactly in both the patients? No. I might be having vasculopathy, he might be having nephropathy, he might be having some other, like, problems. Even tuberous sclerosis also. Okay, there is one more disorder called as a tuberous sclerosis. For example, you can take any autosomal dominant disorder. Now I am having autosomal dominant disorder. The other person is also having the same type of autosomal dominant disorder, but still the symptoms in me and the symptoms in them, they can be different. They can be different. We are having the same diseases. We are having the same genetic mutations, but the clinical features can be different. That is called as the expressivity. Okay. So now I hope you have understood. So the penetrance and expressivity, they are the both different things. Penetrance is how often they will show the disease. Expressivity is how differently they are having the symptoms. Okay. So this is what I want you to know. Now after this, what else you need to know? The most important topic now is autosomal dominant disorder examples. Autosomal dominant disorders. What are the examples of autosomal dominant disorders? One thing you already know. The autosomal dominant disorders, they show incomplete penetrance and variable expressivity. Okay, well and good. Now, what are the examples? Any idea, guys? Any idea, guys? What are the examples of autosomal dominant disorders? Autosomal dominant disorders, they include, let me write here, there are actually 15. I used to remember something like this. Okay, anything which starts with a hereditary, anything which starts with a hereditary, they are almost comes under autosomal dominant. Our family is the most important thing for us, dominant thing for us, our family. So autosomal dominant, anything starts with a family, okay, familial, autosomal dominant, sir. Okay, let me write first here. hereditary. Okay, hereditary. Spherocytosis. Spherocytosis. Hereditary spherocytosis. Next, hereditary cholesterolemia. Okay. Next, hereditary non-polyposis colon cancer, HNPCC, which is also called as Lynch syndrome. Okay, hereditary spherocytosis, hereditary hypercholesterolemia, hereditary non-polyposis colon cancer, colorectal cancer, which is also called as a Lynch syndrome. Okay, important for your exams. Now here, the recent question which was asked in the exam is, in hereditary spherocytosis, which protein defect, can you tell me what is the protein defect in hereditary spherocytosis, where the RBCs will become spherical? RBCs will become spherical, sir. Hereditary spherocytosis. Do you guys have any idea? Hereditary spherocytosis, where RBCs are getting round in shape. The defect is in ankyrin. Okay, MCQ which you should know. Next, so hereditary spherocytosis, Lynch syndrome, hereditary non-polyposis colon cancer. Next, hereditary telangiectasias, and so these are the hereditary things which are completed. Next, I have said you familial. Our family is a dominant thing. Okay, so familial adenomatosis polyposis. Familial adenomatous polyposis. FAP. Next, familial hypercholesterolemias. Okay, familial hypercholesterolemia. Next, I used to remember something like this. Sir, hunting. Okay, hunting is a dominant sport, right? Hunting is a dominant sport. So something like Huntington's disease. Okay. Huntington disease. Next, a very famous MCQ about the kidneys. Okay, about our kidneys. There is one problem which is going to occur to our kidneys, which is autosomal dominant inheritance pattern. Do you know what is that? It starts with the letter AD only. That is AD PKD. Okay, autosomal dominant polycystic kidney disease, which is adult in onset. Okay, adult onset polycystic kidney disease. Adult onset polycystic kidney disease, also called as ADPKD. It is also autosomal dominant disorder. Next, kidney completed. Okay, kidney completed. Ninth, heart related problem. Heart, dominant thing. What? Heart is getting dominant, bigger and bigger. What is that? Hypertrophic cardiomyopathy. Hypertrophic cardiomyopathy, sir. Hypertrophic cardiomyopathy. So it is also following autosomal dominant pattern of inheritance. Now apart from that, okay, now hunting completed. See, simple. Anything which starts with the letter hereditary, hereditary spherocytosis, hereditary hypercholesterolemias, hereditary non-polyposis colorectal cancer, hereditary telangiectasias, or anything starts with the familial also. Okay, so those are all autosomal dominant. Now after that, Huntington disease, hunting, dominant sport. Now autosomal kidney problem, autosomal dominant kidney disease, hypertrophic, big heart. Okay, dominant thing, hypertrophic cardiomyopathy. Now bone problems, two bone problems I want you to know, which are called as osteogenesis imperfecta. Okay, osteogenesis imperfecta, as well as achondroplasia, cartilage problem. Achondroplasia is actually like the cartilage thing, right? So achondroplasia. Okay, achondroplasia. Yes, if you have any notes, you can put. Okay, achondroplasia, osteogenesis imperfecta. Okay, osteogenesis imperfecta. These are the autosomal dominant disorders. They are also following the autosomal dominant inheritance pattern. So apart from that, there are some miscellaneous disorders which you need to by heart. Okay, which you need to by heart for sure. What are they? Leaf-Fermani syndrome. Noonan syndrome. So Leaf-Romani syndrome, Noonan syndrome, and muscular dystonia, muscle. Okay, muscle dystonia, and last and final thing, that is pseudo hypoparathyroidism. In pathology, in the parathyroid topic, we'll discuss about hypoparathyroidism, primary hypoparathyroidism, secondary hypoparathyroidism, pseudo hypoparathyroidism. Okay, pseudo hypoparathyroidism. We'll discuss about them. But right now the point is pseudo hypoparathyroidism. So they are all examples. So 15 completed, sir. Okay. So there are 15 autosomal dominant disorders, which I want you to know is, just don't look at there and just try to answer. What are they? All hereditary things. What are all the hereditary things? Hereditary non-polyposis colorectal cancer, HPC, hereditary non-polyposis colorectal cancer or colon cancer, which is also called as a Lynch syndrome, One thing, hereditary hypercholesterolemias. Okay, next, hereditary spherocytosis, where there is defect in the spectrin, and hereditary, last one, telangiectasias. Telangiectasias means blood vessel problem. Okay, dilation of the blood vessels. Next, familial adenomatous polyposis. Okay, familial adenomatous polyposis. And next, Huntington disease, autosomal dominant polycystic kidney disease, hypertrophic cardiomyopathies, achondroplasia, osteogenesis imperfecta, Leaf-Romani syndrome, Noonan syndrome, muscular dystonia, and last and final thing, it is the everything, hypertrophic cardiomyopathy. All these are the, sorry, last one is not, already I have discussed hypertrophic cardiomyopathy. The last one, pseudo hypoparathyroidism. So these are the autosomal dominant disorders. Now one point which I want you to know is, see, if you are having any of these disorders, autosomal dominant disorders, from when you will get these diseases? From birth or from adolescence? See, these diseases will start to manifest in the body from adolescence or from the adulthood. Okay. So diseases are going to be seen. Disease usually manifest from adolescence. Okay, from the adolescence. These are not childhood onset. Autosomal recessive disorders are childhood onset, but these are not autosomal recessive. These are all autosomal dominant. 15 disorders I gave you. One thing will come in your exam for sure. If you know this 15, more than enough. You need to by heart it. There are many mnemonics which are available in the Google. You can go through them. But see, again you have to remember that mnemonic also. Remembering this 15 names itself, it is difficult. So again remembering a mnemonic is a kind of difficult thing. So just by heart them, sir. Now after autosomal dominant disorders, let me give you examples for autosomal recessive disorders. Autosomal, right? Autosomal recessive disorders. Now there are 15 examples for autosomal dominant. Now you have to know 15 examples for autosomal recessive also. But before telling you, let me tell you something. These are childhood onset. In the next class, I will discuss about the pedigree pattern, how the pedigree questions that will come in your exam. Okay, what is the how to identify the pedigree for autosomal dominant disorders, autosomal recessive, X-linked dominant, X-linked recessive, mitochondrial diseases, how to identify the pedigree. That we will discuss in the next class. In this class, just the examples. Okay, examples part. They are going to be childhood onset disorders. They are going to be there from childhood onwards. Second thing, penetrance. So what about the penetrance? The penetrance is complete. Okay, there are 100 individuals who are having these defective genes. Every person is going to manifest the disease. Every person is going to show the disease. Complete penetrance. It's not like, see, autosomal dominant disorders follow incomplete penetrance, which means even though some individuals are having the defective genes, still they will not manifest the disease. They will not show the disease. But autosomal recessive disorders are not like that. They will have complete penetrance. And expressivity, is also very narrow. Okay, narrow expressivity. Okay, which means if 100 individuals are having the disease, means all 100 individuals will show the same type of symptoms. There is no variability. Sorry, there is no variability, right? All patients will show same symptoms. Okay, so expressivity. Now let's do the examples. Guys, do you know any examples? Do you know any examples, guys, for autosomal recessive disorders? Autosomal recessive disorders examples. Now, see, let me write the examples. Phenylketonuria. I used to remember something like this. Anything which starts with the ends with the letter urea. Phenylketonuria, alkaptonuria, homocystinuria, anything which ends with the word urea, it's autosomal recessive. Okay, so phenyl, ketone, urea. Phenylketonuria, alkaptonuria, homocystinuria. Okay, these are all examples of autosomal recessive disorders. See, out of 15, 3 are completed. Okay, out of 15, 3 are completed. Now, what are the others, sir? Now, let me write one anemia. Which type of anemia? Sickle cell anemia. Sickle cell anemia. Okay, so sickle cell anemia, missense mutation, glutamic acid is replaced with valine. So that example, sir, sickle cell anemia. Next, there is one disorder where a female child will look like a male. A female child is going to look like a male, sir. That is female pseudo hermaphroditism. What is that condition? Congenital adrenal hyperplasia. So congenital adrenal hyperplasia, deficiency of enzyme 21 hydroxylase and 11 beta hydroxylase. So that syndrome, sir. Okay, congenital adrenal hyperplasia. See, important points about this autosomal dominant disorders. For example, important points what you need to know regarding congenital adrenal hyperplasia will be discussed in the next video. Okay, even some important autosomal dominant. Okay, let me write one important thing I forgot here. In autosomal dominant, add one more thing. 16th, Marfan syndrome. Marfan syndrome, MCQ. This is very important, sir. Marfan syndrome. Next, 17th, neurofibromatosis. Neurofibromatosis. Okay, Marfan syndrome, neurofibromatosis also follows the autosomal dominant inheritance pattern. Okay, next, let's come to the autosomal recessive. What we have completed: phenylketonuria, alkaptonuria, homocystinuria, sickle cell anemia, congenital adrenal hyperplasia. Next, Friedreich ataxia. Okay, Friedreich ataxia. Next, any other disorder, do you know, where there is excessive deposition of copper in the body? There is this one disorder where there is excessive deposition of copper in the body, which is called as Wilson disease. Wilson's disease. And excessive deposition of iron in the body, that's the hemochromatosis. Okay, hemochromatosis, sir. So Wilson's disease, important points about the Wilson's disease, that's celluroplasmin deficiency is there, the Kaiser-Fleischer rings will be seen in the eyes, and hemochromatosis, excessive iron deposition in the body, the iron is stained with the pearls Prussian blue. See, as I'm having a little cold now, I'm not going in details about the important genetic disorders. In the next class, one genetic disorder I will take. Marfan syndrome, important points. Okay, look, Marfan syndrome, what are the important points which you need to know? Neurofibromatosis, what are the important points which you need to know? Okay, and let me add one more autosomal dominant disorder, that is tuberosclerosis. Okay, tuberosclerosis. This is also autosomal dominant disorder. So I will discuss about this autosomal dominant disorders, important autosomal dominant disorders which you need to know for your exams. Oh, this is the important MCQ about the Marfan syndrome. This is the important MCQ about the phenylketonuria, something like that. Okay, so phenylketonuria, alkaptonuria, homocystinuria, important points are there. Autosomal recessive disorders I will discuss in the next class, sir. Okay, because I'm little irritation is there in the nose, so I will complete it quickly. So hemochromatosis, Wilson's disease completed. Now after that, see all the inborn errors of metabolism, like the lysosomal storage diseases, lysosomal storage. You know, right? Niemann-Pick disease, Gaucher's disease. Okay, those diseases, lysosomal storage disorders, and even glycogen storage disorders. Lysosomal storage disorders and glycogen storage disorders are all these disorders, lysosomal storage disorders, glycogen storage disorders, they are all examples of autosomal recessive disorders. Now next, one more thing, cystic fibrosis. Cystic fibrosis, you know it, right? CFTR gene mutation, thick mucuses are going to be produced in the body, leading to the obstruction of the ducts. So cystic fibrosis leading to pancreatitis, acute pancreatitis, and even bronchiectasis in the lungs. So cystic fibrosis. Next, ataxia, telangiectasia. Okay, next Fanconi's anemia. See, these are all examples. Okay, these are all examples of autosomal recessive disorders. Any other examples? Yes, xeroderma, pigmentosa. Next, a syndrome called as a Bloom syndrome. So these are examples which you need to know for your exam. If you know this much, that's more than enough, sir. Okay, so like, see, just recap. What are the examples of autosomal recessive disorders? The examples of the autosomal recessive disorders are phenylketonuria, alkaptonuria, homocystinuria, sickle cell anemia, congenital adrenal hyperplasia. Now, one important point is albinism, right? Albinism. Okay, so albinism, one more example which is following the autosomal recessive inheritance pattern. Next, Friedreich's ataxia, Wilson's disease, hemochromatosis, all lysosomal storage disorders. Exceptions are there. Exceptions are there. Hunters is an exception. Okay, Hunters is an exception. We'll discuss later. Lysosomal storage disorders, glycogen storage disorders, cystic fibrosis, ataxia telangiectasias, Fanconi's anemia, xeroderma pigmentosa, Bloom syndrome. These are all examples of autosomal recessive disorders. Now, one more thing, alpha-1 antitrypsin deficiency. Alpha-1 antitrypsin deficiency leading to emphysema. Okay, leading to emphysema. So this is also an example of autosomal recessive disorders. Important points about autosomal dominant, autosomal recessive disorders in detail will be discussed in the next video. So today let's stop here. Okay, examples of autosomal dominant and examples of autosomal recessive. We'll discuss much more important points in the next class. The X-linked dominant disorders and the X-linked recessive disorders, and also the pedigrees, the mitochondrial diseases and non-Mendelian disorders will be discussed in the subsequent video. Okay guys, today I am a little nauseous, a little irritation is there. So let's stop the class till here for today and we'll continue in the next class. Hope the class is helpful. See you next time. Thank you.